Next technique, it is northern blotting. Northern blotting, it is used for the analysis of RNA molecule, and southern blotting, as I already explained, it is used to analyze DNA molecules. Now, northern blotting, it is a technique or method that is used to detect RNA fragments from a mixture of RNA molecules. And we can analyze RNA from any cell type, tissue, or any sample. So here, this diagram showed the overview of northern blotting. The process is more or less similar to southern blotting that was used for DNA analysis. So here, the RNA samples or fragments, they were first separated by gel electrophoresis, and of course, they are bounded in the next step to nitrocellulose membrane or any other membrane. And after blotting, then the membrane, it is placed in a solution of radio-labeled isotope here, and finally it is placed in contact with X-ray film, and the hybridization, it can be detected by autoradiography. This is another diagram that indicated the RNA detection by northern blotting. So here RNA, it is extracted from any sample or cell type, and then it is separated on gel electrophoresis so that different fragments of RNA, they are well separated from each other on the basis of their molecular weight, and then in the next step they are immobilized on the membrane for fixation process, and after fixation, nitrocellulose membrane, then it is placed in a solution of probe here, and when hybridization process is there due to complementary basis, then in the final step the hybridization can be detected by autoradiography. So this is an overview of RNA detection by northern blotting.